Hey guys, welcome to my channel. So today in this video we are going to talk about nutrition in autotrophs. So let's get started. Food is needed by all living organisms mainly for growth and repair. Several organisms need food to maintain their body temperature as well. A variety of substances are taken as food by every organism from single cellular organisms like amoeba to complex multicellular organisms like the human body. Even within the human body, the cells require a wide variety of substances as food to carry out their functions. The mode of acquiring food varies from organism to organism. What are autotrophs and how do they get their food? So answer is we all know that. So answer is here. We all know that the autotrophs are the only organisms which can prepare their own food by using the light energy to synthesize the chemical compounds. They acquire water and mineral salts from the soil as well as certain gases from the air. By this, raw materials are capable of preparing complex compounds like carbohydrate, proteins, and lipids from very simple substances. These compounds produced by autotrophic plants are utilized for providing energy to the most of the living organisms. Most of the food that we eat are obtained from plants. Even if we depend on animal products, we would find that those animals depend on plants for their food. So next question is it, how do plants carry out these life processes? To answer all these questions, scientists have worked for centuries to find out how plants carry out these life processes. Among all the life process, the process of photosynthesis make the plants the universal food providers for the living organism. Research on plant life processes have really started long ago. Actually, experimentation to know about plant needs have been recorded from 17th century. A scientist named Van Helmont had established the role of water by an experiment conducted by him for a period of 5 years, and it took the mankind another 300 years to reach the present definition of photosynthesis. Actually, what is this photosynthesis? Photosynthesis is a process by which plants containing the green pigment chlorophyll build up complex organic molecules from using simple inorganic ones, using the sunlight as an energy source. A scientist named C.B. Van in 1931 had formulated a simple equation to indicate the process photosynthesis. His opinion was for each molecule of carbohydrate formation, one molecule of carbon dioxide and two molecules of water are required. In this process, along with carbohydrate, one molecule of oxygen and water are produced. When C.B. Van worked on purple cells for bacteria, he came to know that the light plays a specific role in photosynthesis. Instead of H2O, he used H2S as a starting material. Here, no oxygen is liberated during photosynthesis. Instead, it is sulfur. Later, he envisioned similar process of photosynthesis in plants. The above equation was proposed when glucose is observed to be a product in photosynthesis. And there is a fact. It is known that plants synthesize carbohydrates, the smaller, simpler ones first, and from them the more complex ones like starch and cellulose. Plants are also capable of synthesizing all other compounds like proteins, fats, etc. So another question here we have. Can we state that photosynthesis is the basic energy source for the most of the living world? Why or why not? Photosynthesis, the process by which green plants and blue-green algae use the energy of light to convert carbon dioxide and water into simple sugar glucose. In so doing, Photosynthesis provides the basic energy source for virtually all organisms. Photosynthesis is important to live organism because it is the source of the organism in the atmosphere. Without photosynthesis, the carbon cycle could not occur, oxygen requiring life wouldn't survive. The process provides the initial organic carbon source in the form of glucose and oxygen as the byproduct. So, let's know about the activity 1. So, the activity 1 name is presence of starch in the leaves. Aim. Our aim is to prove that carbohydrate is present in the leaf. Materials required. Wide mouth or beaker with water, test tube, asbestos gauge, tripod stand, Bunsen burner, petri dish, narrow leaf. Chemicals required. Methylated spirit, iodine solution. Procedure. Take a wide mouthed beaker with water. Add few ml of methylated spirit and narrow leaf to the test tube and place it in the beaker with water, and setup should be kept on the asbestos gauge. The entire setup again should be kept on the tripod stand. Heat is applied to the beaker with water through asbestos gauge with the help of Bunsen burner. The test tube with methylated spirit and the narrow leaf should get boiled. After boiling the leaf and methylated spirit, lift it out with the help of tongs. When the leaf is treated with methylated spirit, it loses all the green color and turns to pale color. Now take the leaf into petri dish and add few drops of iodine solution to the leaf. So iodine is a reagent solution which defines the presence of carbohydrate, which means a starch or glucose in the leaf. Observation. When the leaf is treated with iodine solution, the pale colored leaf turns into dark blue or black blue. The dark blue or black blue color indicates the presence of carbohydrate, which means the starch or glucose in the leaf. Conclusion. By this experiment, we can conclude that starch is present in the leaves. Okay, next video is about the factors essential for the process of photosynthesis. What are the materials that you think would be essential for the photosynthesis of carbohydrates in the process of photosynthesis? So, hint is see the equation proposed by Van. Okay, let's go. Yeah, this is the equation produced by Van, right? So what are the raw materials? So let's start raw materials first. Carbohydrate, sorry, carbon dioxide and water and chlorophyll is required. Then in the presence of light, we will get the products as glucose, water and oxygen. Okay. Yeah. Yeah. And do you think the equation tells us about all the materials involved? So to answer, let's study how scientists experimented to find out about some of the materials required for the process of photosynthesis. Water and photosynthesis. So, when we are in 7th, we have studied that von Helmont found that water is essential for the increase of plant mass. Then he didn't know about the photosynthesis then. Later, it was found that increase in plant body mass or material occurred due to the process of photosynthesis. Air and photosynthesis. Okay, now let's discuss an experiment on photosynthesis. This one helps us to find out about the role of the role of air in the process of photosynthesis. It is interesting to learn about experiment, which is one of the several milestones in the gradual development of our understanding of photosynthesis. Joseph Presley. In 1770, performed a series of experiments that reveal the essential role of air and the growth of green plants. We can really we can recall oxygen was discovered by Presley in 1774. The name oxygen was later coined by Lavoisier in the year 1775. Presley observed that a candle burning in a closed bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space of the bell jar. He concluded that burning candle or an animal would somehow damage the air. When he placed a mint plant in the same bell jars, he found that the mouse stayed alive and the candle, when lighted from outside, continued burning in the presence of the mint plant. Presley hypothesized as follows: plants restored the air, what breathing animals and burning candles removed. So this was the image of the experiment. So Presley's experiment. Do you find any relation between candle, rat, and mint plant? Discuss. Presley's experiment confined that gaseous exchange was going on, and plants were giving out a gas that supported burning and was essential for survival of animals. But how do plants take in air and utilize carbon dioxide from photosynthesis and oxygen for respiration? How do they make their choice? So answer is massive amounts of gases exchange occur through the stomata as long as they are open. While plants also carry on gaseous exchange through loose tissues in the stem, roots, etc. It is actually at the level of organelles involved in the process of photosynthesis and respiration that the choice of the gas required is made. Activity 2. Carbon dioxide is necessary for photosynthesis. Mole's half-leaf experiment. We need a discharge plant to start with. We need to keep the plant in the dark for nearly 3 days to remove the starch. By placing the plant in the dark, the starch is removed from the leaf. Arrange the apparatus as shown in the figure. Take a wide-mouthed beaker, transparent bottle. Materials required are potted plant, spill cork, wide-mouthed bottle, potassium hydroxide and iodine. Put potassium hydroxide petlets or potassium hydroxide solution in the bottle. Potassium hydroxide absorbs carbon dioxide. Insert filtered cork in the mouth of the bottle. Insert one of the leaf of discharge plant into transparent bottle containing potassium hydroxide pellets. Potassium hydroxide solution. Leave the plant in sunlight. After a few hours, test this leaf and any other leaf of this plant for presence of starch as mentioned in the activity 1. So how we will test it out? We will take the leaf into the petri dish and we will add few drops of iodine. And same, if the leaf turns to blue black or dark blue or black, uh, it represents that the carbohydrate is present in the leaf. Okay. The leaf part which is exposed to the atmospheric air and light becomes bluish black, and the one inside the bottle containing potassium hydroxide or KOH. So this KOH absorbs the carbon dioxide from the leaf. So this carbon dioxide in the bottle remains colorless. This proves that carbon dioxide is necessary for the photosynthesis. So why was the plant kept in the dark and then in sunlight? So we kept in the dark because to discharge it, and then sunlight for the for the formation of photosynthesis. Okay. Why did we test two leaves in this experiment? Uh, we tested because one is kept in the transparent bottle in which is with KOH, right, which absorbs the carbon dioxide, and one was exposed to sunlight completely. So to make notify which any changes both of in the leaves. Okay, so this is the reason we tested both the leaves in this experiment. So next, light and photosynthesis. In Presley's time, scientists did not understand about energy, but later on much was discovered about it. If energy is released when carbon dioxide and water is formed by combining oxygen with carbon and hydrogen. Then what about rivers? What about forming oxygen again and putting it back into the air? Eventually, scientists learned that energy situation would also reverse. Oxygen formation would use up energy. That means if plants form oxygen, they have to get energy to make it possible. So, where did the energy actually come from? So, to answer this, adapt scientist John Ingenhouse studied that the way which plants form oxygen. In 1779, he noticed that it happened only in the presence of light. In an experiment with the aquatic plant hydrilla, he observed that the bright sunlight, small bubbles formed around, in the bright sunlight, small bubbles form around the green parts, while in the dark, they did not form. He also found that the gas present in the bubbles was oxygen. It was further confirmed when Engelmann, in the early 20th century, Ingenhousz detected the point of maximum rate of photosynthesis. He used a strand of algae and exposed it to different colors of light, the colors that we see in a rainbow. Then he used the oxygen sensitive bacteria and found that they crowd around areas illuminated with red and blue rays of sunlight. This led to more studies on the effect of light on photosynthesis, the role of different colored compounds called pigments in plants, and the utilization of light energy. Lab activity. So we need to arrange the apparatus as shown in the figure. So first we need the materials. So what we need is oxygen. Sorry. We need the apparatus are required are test tube, beaker, water, funnel, hydrolytic plant. So you can take any other aquatic plant, but the plant should completely submerged into the water. So example is hydrolytic. So we have taken hydrolytic. So let's one. Place some water plant like Edula or Hydrilla in a short stemmed funnel and keep it in a beaker. Then pour the water in the beaker. Invert a test tube full of water over the stem of the funnel. Ensure that the level of water in the beaker is above the level of stem of the inverted funnel. Place one of the apparatus in the sun. So we have to make these two identical sets, right? So one should be placed in the sun and one should be placed in the dark. They should be placed for at least for 2 to 3 hours. Okay, you should see that in place of water there is a air that fills in the set up kept in sun. It is actually a gas that will be collected in a test tube. Then observe the other setup kept in dark. Is there any difference in the amount of gas collected? Okay, there would be a difference, definitely a difference, because photosynthesis takes place only when there is a sunlight. So we have kept in the dark. So there is no chance to form photosynthesis actually. So now what is the gas? So now we are going to test this gas. So now test the gas in a test tube by inserting a glowing matchstick or incense stick which would burst into flames. This shows the presence of oxygen. We know that the oxygen is the only gas which supports the combustion, right? So now, what are the precautions do we need to take while removing the test tube from the vehicle? So we need to take that precaution that is, we should take, we should be careful so that the oxygen or the gas should not escape actually. Okay. So activity 3. Sunlight is necessary to form starch in the green plants. Now, take the potted plant with discharged leaves. Remember the process of discharging leaves mentioned in the activity 2. So, in activity 2, which is more safe experiment, in that activity, we kept the plant in dark for 3 days to discharge. Okay. So, now for this activity also, we should do the same. Then, cover one of its leaf with black paper on which a design cut. Fix the paper on the leaf in such a manner that light does not enter the dark part. Place the spotted plant in sunlight. After a few hours of the exposure to the bright sunlight, test the leaf which is covered by black paper for the presence of starch. Which part of the leaf turns blue black? Actually, the leaf which is exposed to sunlight. Okay. What about the remaining part? So, which is covered by the black paper doesn't turn to bluish black. It turns to pale color. Observe the color of the leaf stained with iodine. Can you tell why it is stained differently? It will be observed that the only parts of the leaf which could get light through the cut out design turns blue black, showing the presence of starch. Okay. So next, chlorophyll and photosynthesis. Ingenhorn wanted to find out more about photosynthesis and carried out several other experiments. He proposed that only green plant parts could carry out the process of photosynthesis. What about plants having colored leaves? How is it that new leaves which look dark red in color in several plants turn green? Do plants having red or reddish or yellowish leaves also carry photosynthesis? What made plants carry out the photosynthesis while even green colored animals like some birds couldn't? Questions like these remain challenges until scientists could isolate the green colored substances from plant parts and study their nature. Establishment of Ingersoll's proposition came after several experiments till the mid-20th century, when scientists could also locate the site of photosynthesis and even isolate it. Around 4 decades after Ingenhouse's proposition, scientists could only isolate the green substances to observe its nature and find out where the photosynthesis could be carried out with it. This had become possible in the year 1817 due to the work of two scientists, Peltier and Cavendish, who obtained an extract of the green-colored substances and named it chlorophyll, meaning green leaf. It was also found that pigments, in addition to chlorophyll, carotids, and psychobilins, could also aid in the process of photosynthesis by passing on the energy of sunlight trapped by them to chlorophyll. Actually, where does this photosynthesis take place? Where chlorophyll and other pigments present in the plant? Try to name some parts where you think photosynthesis occur. Do you think the new reddish leaves of plants also carry the photosynthesis? What could be the reason for the color? So answer all this question. This is the paragraph which has answer of all this question. The exact location of photosynthetic part or a part containing chlorophyll was not known until another 6 decades after Peltier and Cavendish discovered chlorophyll. It was believed to be spread in the cell of green plant parts. In 1883, Julius Sachs, one Sachs, Julius Julius von Sachs observed that chlorophyll in plant cells is not spread throughout the entire cell. It is rather found in the organelles within the cell. Such organelles are named as chloroplasts. These are present in large number in the cells, around 40 to 100 of parts, like the stomatal gut cells and ground tissues of plant. So this is the tears of the leaf. Tears of the leaf showing chloroplast in palisade spongy parenchyma. What makes the chloroplast appear completely different from other cell organelles? So there is a fact again. If a cell breaks up, the chloroplast also break into pieces. So it becomes a very difficult task to isolate them, to to study the different steps of photosynthesis. It was not until 1954 that Daniel I. Arnon was able to break up plant cells so gently that whole chloroplast could be obtained that could be carried through the photosynthesis. So let's continue. It was being found that the chloroplast is a membranous structure consisting of three membranes. The third layer forms staked sac-like structures called as grana. It is believed that to be a site for trapping the solar energy. The intermediary fluid-filled portion is called as stroma. It is believed to be responsible for enzymatic reaction leading to the synthesis of glucose, which in turn join together to form starch. Substances found in chloroplasts which capture sunlight are called photosynthetic pigments. There are several types of photosynthetic pigments involved in the process to produce organic molecules like glucose in the plants. Chlorophyll is such a pigment which contains one atom of magnesium. It is similar in structure to the heme of hemoglobin. Heme of sorry, heme of hemoglobin. The iron containing red pigment that transports oxygen in the blood. Oxygen in blood. Two major kinds of chlorophyll chlorophylls are associated with thylakoid membranes. Chlorophyll A is a blue-green in color and the chlorophyll B is yellow-green in color. Around 250 to 400 pigment molecules are grouped as the sunlight harvesting complex of photosynthetic unit in each thylakoid. Such innumerable units function together in chloroplast of green plants in the process of photosynthesis. During the photosynthesis, several events occur in the chloroplast of some of them are conversion of light energy to chemical energy, splitting of water molecule, which is known as photolysis of water, reduction of carbon dioxide to carbohydrates. Light is required to initiate several events while several may continue even in the absence of it. What would that would mean once light energy has been captured, it can help reactions to continue even in the dark. Light dependent events or reactions are called light reaction and it has been found that to take place in grana while the rest are called light independent or dark reactions and they occur in the stroma. Light reactions. This light reactions takes place in the daytime. It takes place in the grana of the chloroplast. The chlorophyll will expose to sunlight and get activated and absorbs photons. Photons means the unit of light energy. Using the solar energy, the water molecule will split into positive ion, which are H plus, and the negative ion, which is OH minus. This is known as Hill reaction. OH ions will undergo many reactions and forms water and O2. Water is used by plants and this O2 will be released into the atmosphere. ATP and NADPH are liberated and stored in mitochondria. ATP, NADPH, and O2 are known as assimilatory powers. These light-dependent reaction is also known as photochemical phase. So next is about the light-independent reaction. H plus protons move outside the thylakoid and cause ADP and P to become ATP. Because they run heavier concentration of H plus protons in the thylakoid membrane than in the stroma. So the H plus will move out into the stroma. As the H plus protons move into stroma, they release energy which is converted into chemical energy stored in ATP. Light independent reaction or Kelvin cycle or dark reaction. The Kelvin cycle uses the energy in ATP and NADPH made during the light reaction to fix carbon into an organic compound called PGAL. Takes place in stroma. Carbon dioxide diffuses into cell and combines with IUBP to form PGA. Molecules of IUBP, 5-carbon sugar, are present in the cell. One carbon from CO2 attaches to each IUBP. This 6-carbon is unstable and immediately splits into half. This leaves 3-carbon structures called PGA ribulose biphosphate phosphoglycerate. PGA is changed into PGAL. PGA interacts with ATP and NADPH from sunlight reactions. So RxNS means reactions. So this ATP and NADPH resulting in PGA, new 3-carbon structure. The ATP and NADPH are returned to the light reaction as ADP and NADP plus and a phosphate group to be reused. One PGAL leaves the cycle to become glucose uh, phosphoglycerol 8. Okay, phosphoglycerol 8. The PGAL is converted back into IUBP. The remaining PGAL 3 carbons rearranged into IUBP 5 carbons with ATP's energy. Cycle starts all over again. Glucose is not made during photosynthesis. PGAL molecules are made which are built into carbohydrates just after photosynthesis ends. So this is a mind map of photosynthesis. So requirements of chlorophyll, carbon dioxide, sunlight, water occurs inside the chloroplast. This process consists of light reaction and dark reaction. Rate can be affected by light intensity and carbon dioxide concentration. Products are glucose and oxygen. Glucose forms in cell, provides energy. Glucose are converted into amino acid, sucrates, starch, cellulose, fats. So in this video we have learned about the nutrition and autotrophs, right? So in another video let's learn about the heterotrophic nutrition. So until then, bye bye. If you hadn't subscribed my channel, please remember to subscribe my channel. See you later. Bye bye.